Share the screen. All right, there we go. So the question is on the case studies of the lab, and this is lab 4, is that correct?
Yes.
Lab 4. If you look at the case study 1, you can see that this area of the lung, something looks wrong. The lung should be very dark like this. In fact, there's a little bit of light there and a little bit of light here and a little bit there. And that means the person has something in their lungs. It's probably mucus, but we don't know what else might be in the lungs. And then the person gives sputum. Do you know what that is? A spit culture.
Yeah, I got that.
AFB culture for acid-fast bacteria. And then they did the Zeal-Nielsen stain on it. What do you notice in the Zeal-Nielsen stain?
There is positive and negative bacteria on it.
Okay.
And it's
which ones are the positive?
The positive would be the blue, and then wait, no, for the acid stain, the positive is
okay, just figure that out.
Right.
You do get the idea that there are both positive and negative. And
right.
It's very normal when somebody is sick that they have some bacteria which will be causing the sickness, in this case, the positive ones, and some that are negative, which are just the person's normal bacterial flora, which I shouldn't call it flora, I should call it microbacteria. Microcosm, what is that word? Mycobacteria, whatever the name is, I've forgotten the microflora.
Microbiota?
Microbiota, there we go. I was trying to remember the new name. Flora is the old name, which I learned when I went through microbiology, and I knew I was saying the wrong thing. All right, so you know that there's positive and negative in here. And what does that mean?
I believe it means there's a pathogen in there that's causing the illness, and you
but
have to figure out
what is the type of the pathogen? What does the acid-fast pathogen mean?
Is it endospore?
Look up the Zeal-Nielsen stain. You haven't done that. And then that should help you with case study 1. Meaning this stain is looking specifically for one type of pathogen, and it's the acid-fast ones. And so what do they cause? And then that should tell you what this patient has. Okay?
Okay.
All right.
Was that in the module and I just missed it?
Yeah, look up. We did talk about acid-fast bacteria and then the diseases that acid-fast bacteria cause. Okay. All right. Case study 2 is a 64-year-old diabetic patient. This isn't terribly important and isn't covered in the module, but he's admitted to the hospital with soft tissue necrosis. And you look at that, you blow that up, and the foot looks very, very bad. That's really bad necrosis. In fact, if you look there and there, it looks like this tissue is dying. Okay. They did a routine culture and then did the gram stain, and there's the bacteria in the gram stain. What is this bacteria? You don't know the species, but you can tell me what type it is. Are you there?
Sorry, I'm not sure what you mean. You mean like coccus or bacillus? Is that what you mean?
It's going to be either coccus or bacillus. Yes, that's correct. And I'm asking you what we're seeing here in the gram stain.
The bacillus is positive.
It's gram positive bacilli. These cells here are probably the patient's own cells. Surprisingly, we don't see any other bacteria. And usually we'd see some bacteria from the patient's normal flora. But what they may have done was done a biopsy inside the patient, and the normal flora isn't inside his skin, underneath the skin, but the bacteria causing the problem is. Okay, so it's a gram-positive bacilli. That should help you. And then we have another stain here, and it tells you it's the Schaefer-Fulton stain. What's the Schaefer-Fulton stain give you?
Positive as well, right? Because acid, fast, base,
Positive,
and the
but
gram is
positive for what?
opposite.
It is positive, but what's it positive for?
Well, that's what I have to look up, right?
Yeah,
Because
you have to look that up. And then there's one other hint, and it says the bacteria are only growing anaerobically, so that these bacilli will not grow in the air. So they didn't get this bacteria from the bottom of his foot. They must have done a biopsy inside the foot to get this bacteria, because it only grows anaerobically. So that gives you another hint. And once you figure that hint out with the Schaefer-Fulton stain, that will tell you where to look in the lab module. And then you should be able to identify the species of bacteria that's causing this problem. Okay?
okay,
Questions?
I think I got it.
All right.
No, I was just making sure that there's some something to read about it in the module, and it's not just Google-based answers.
The gram-positive rods, probably the anaerobic bacteria, let me stop my share, is in the lab module. I think what happened to the, I think the Schaefer-Fulton stain is in the lab module, but you might have to Google that one. And
Okay.
look, you're not seeing the screen now, right? Let me look and see.
No,
I know
I'm
the
not.
species that's causing this is, it's not in the worksheet, it's in the lab module. Let me get the lab module open.
So acid fast staining stains bacteria such as Mycobacterium and Nocardia.
Uh-huh.
Is that what you mean? Okay.
Uh-huh.
Oh, tuberculosis. I wondered, but I was trying to look up pictures to confirm what the bacteria look like, and I had a hard time getting the exact
Oh no, that
same.